To talk about the Science Breviary article published in 2008 by McDonald and colleagues. This article, I think, entitled Bioactive Contaminants Which Leach from Disposable Laboratory Plasticware, is of particular interest to all of us as laboratory researchers. And the reason being is that it provides alarming evidence that there are contaminants in commonly used laboratory plasticware that could have significant impact on your research. Now, I think we're all quite familiar with centrifuge tubes such as these. If you're like me, you probably assume that freshly autoclaved, unused tubes are completely clean, completely free of any contaminants, and that there's no confounding variables going on in your experiments. And ideally, that would be the case. I mean, ideally, the only variables in your experiments should be the reagents that you put in there. However, as reported by this publication, it turns out that may not actually be the case. There may actually be contaminants in these centrifuge tubes which significantly inhibit many biochemical enzymes. So the story begins with the authors of this paper studying human monoamine oxidase B. And they keep coming up with frustratingly inconsistent results. These researchers were fortunately clever enough to recognize that monoamine oxidase B can recognize many xenobiotic substances. And so they set out to find what the source of these xenobiotic contaminations might be. The methodology was quite simple. They used common laboratory solvents, water or DMSO, and they rinsed microcentrifuge tubes and pipette tips and collected the solvents after rinsing. The idea is that rinsing these laboratory plasticware items would solubilize any contaminants found in the residues, and they then collected these solvents and used them in biochemical assays. So they had found that these collected solvents actually had profound inhibitory effects in many enzyme assays. So here in figure 1, on the y-axis, we'll be measuring the relative activity of human monoamine oxidase B, and it's going to be normalized to a control sample of solvents run through control glass vials that are clean, there's no residue. On the x-axis, we're looking at various samples of either water or DMSO that have been run through centrifuge tubes of various volumes, ranging from 500 microliters to 15 milliliters, using 2 different vendors. And right away we can draw a few conclusions. We can see that there is indeed some inhibition going on with different centrifuge tubes, and in some cases there's even potentiation. Secondly, we conclude that using DMSO gives you a more profound effect than using water. And thirdly, we can also see that this effect is not solely contributed to a single vendor. We can't conclude that we should forego buying centrifuge tubes from vendor A and instead buy from vendor B. Instead, we see that this sort of inhibition is quite prevalent, and we can probably assume that this sort of inhibition is happening in our labs using our centrifuge tubes. Now, is this inhibition something that is only relevant to monoamine oxidase B, or can it be translated to other areas of interest? Next, the researchers translated this to another protein of interest, the GABA-A channel. Here we're looking at a specific binding assay in which they've taken their solvent and they've rinsed it through Eppendorf tubes 1 time or 3 times, and they're comparing this to the same solvent run through glass vials. And we can see that after running solvents through Eppendorf tubes 3 times, you see a very profound, significant effect of reduction in specific binding. On the right side, we're looking at a competition binding assay, in which they have done 2 parallel experiments, one in plastic microcentrifuge tubes and a second in glass vials. And you can see a suppression of the maximum binding by about 20%. And that's quite profound. This implies that if the 2 different researchers or 2 labs doing identical experiments can come up with significantly different results. So to conclude, we see that commonly used disposable laboratory plasticware contain bioactive chemical contaminants. Secondly, we see that the choice of manufacturer, the choice of solvent, both of these may significantly impact the extent of contaminant mobilization. And thirdly, we see that leached contaminants can skew experimental results. Thank you for your attention.